And now I wish to welcome Dr. Anja Krivograd. She is a molecular biologist focused on the science of aging, including genetics of aging, clinical research, and holistic health. Her doctoral work at Free University of Berlin and German Center for Neurodegenerative Diseases explored anti-aging effects of spermidine and protein restriction. We are welcome to have her here.
Thank you for having me. It's a privilege to be able to share some of my research experience with you today. In today's talk, I'm going to show you the health benefits of one natural substance called spermidine and one nutritional intervention, protein restriction. This is a story involving research on Drosophila, on fruit flies, and a human clinical trial. So, but not to be too mechanistic and academic, let us also have a look at the aging research a little bit from the distance. As we know, aging is not only a biomedical concern, but it also crucially shapes the societal dynamics. That's why it's important to acknowledge today a little bit on how we started in aging research, how it's going, and what we can expect from the future of aging research. In the very end, I'm going to give you some excerpts from my research on fruit flies, and I will also talk about some excerpts of spermidine's health effects on immune system rejuvenation in the clinical trial called ImmuneAge. Now, we don't know each other, so to kick this off, let me make this a little bit personal. Considering myself, those are likely my remaining weeks alive. Now, I have two questions for you. First of all, if you were guaranteed and given the perfect health until your very last day, tell me with a raise of hands, how long is it that you would like to live? So, raise your hands, Team A. Okay. So how about B?
Yeah.
Okay. And do we have any C's in the audience? All right. Yeah. And okay. But then, on the other hand, considering your current health habits, your family health history, and, well, the overall feeling, how long is it that you think you will actually live? Again, go A. Okay, good. So where are B's? Mm-hmm. And do we have any super centenarian in the audience? Of course. I'm asking this because I couldn't find any European-based study, but when over 2,000 Americans were asked, it's interesting that the sweet spot for desired lifespan revolves around 90 years old, regardless of the age group. Interestingly, the answers for the treatments, the future treatments, which will enable people to live until 120, are considered to be a bad thing for society in the majority of cases, and most people doubt that we will achieve this within the next 30-ish years. Now, I wanted to draw together some of the longevity, the current statistics on the global average lifespan. And what I see is that, or what is the fact is that basically from the current highest global average lifespan of 85 to the current global lowest lifespan of 54, there is still quite a big age gap of about 30-ish years. In today's day and time, this is quite striking. It is no surprise that females globally still outlive males with some sort of like an average gap of 5 years. And yeah, here are the trends in the global lifespan, in the global average lifespan. From 1900s, the global average of 33 throughout 1950s and 2000s, we have achieved quite significant improvement. The current global average lifespan expectancy is 73 years old, and we have over 700,000 centenarians. Now, this curve kind of coincides with some of the milestones in the aging research. In early 1900s, the first intervention, which was the first intervention to be shown to extend lifespan significantly in rodents, was caloric restriction. And from here, the first breakthrough happened with the identification of the first single gene mutation that extensively expanded lifespan in worms. This was the H1 gene. And then the mutation of the DAF2 gene, which is associated with the insulin IGF-1 pathway, as the first conserved pathway between species that regulates lifespan. So this kind of marked the era of mechanistic research, and with the onset of the pharmaceutical era through years of 2000s, this kicked off clinical trials on aging in the humans, and it was important because it showed us that we can extend lifespan also in the later age, that it's never too late. The future likely brings the so-called precision medicine, which will enable us to do basically individual screenings and real-time optimizations of health based on the biomarkers. And it is much more focused on preventive rather than reactive. So, as I already kind of illustrated, the mechanistic research is the backbone of it all. Especially since because the predictions show that until 2050s, the 65-year-olds will globally outnumber under 15-year-olds for the first time in human history. So it is important that we start adding life to years, not just years to life, by maintaining and preserving the cognitive and physical capacities into the late age. This will, of course, require a different scientific and medical approach. We can't just keep targeting single diseases and single symptoms and diseases, but we need to, with this precision medicine, because the targeting just like single symptoms is basically very costly, and not to mention other disadvantages of that. So the new era of medicine and science is going to really enable this personalized approach to preventive treatments and to proactively and not reactively take care of aging. And practically, this is going to make a big shift in how we conduct science in the future. What is important also for the population at large, I would emphasize, is that we discern this scientific-based facts from the current marketing hypes on aging. So when it comes to factors which determine our lifespan and our health span, the good or the bad news is that lifestyle still has a foundational role in prolonging or in deciding on the health outcomes. So it is estimated that with lifestyle factors, such as we know them already, kind of are pretty familiar to all of you, we can decide for up to 80% of the outcomes, while your genetic faith only contributes up to about 30%. So in my former lab in Berlin, we focused on the aspects of nutritional interventions, where we studied the effects of spermidine supplementation and protein restriction in fruit flies. So together with my colleagues and external collaborators, we conducted a clinical trial, ImmuneAge, in which we focused, as the full name suggests, the effects of short-term spermidine supplementation on functional reprogramming of the immune system in young and old adults. So We took two very health homogeneous groups, the young population of 25 to 40 and the elderly of 70 to 19 years old, who were equally randomized into placebo or spermidine group, receiving 6 milligrams of spermidine daily over the course of 20 days. This was by then the highest used dose in clinical trials, and according to the estimates for developed countries, increases amounts to about 60% of increase from the average spermidine consumption through nutrition. And so from blood, from the plasma serum and PBMCs, we drew several measurements. So we basically wanted to identify, first of all, study the mechanisms in PBMCs that are regulating aging, so the key mechanisms such as autophagy and hyposination, which I will explain in a second. Then we wanted to identify blood biomarkers such as polyamine, quantified polyamines, or inflammatory markers that could potentially be diagnostic in evaluating the aging progression of immune system or the success of spermidine supplementation. And as the primary readout, we wanted to look into the functional, so assess spermidine as an adjuvant for mediating functional improvements in the immune system in the elderly. Today we will go into some excerpts, but for the time constricting reasons, I cannot show you the whole study. First of all, to look into the spermidine-related results, what we saw, let me introduce this compound to you a little bit. So what is spermidine? It's basically, so if you imagine spermidine belongs to a different family of molecules, polyamines, and it is a substance that is naturally present in various food sources. We eat it every day. So this is the major source of spermidine is coming through diet and per content, so content per volume, it is the highest represented in wheat germs, in fermented soybeans, and a very high content we also find in, for example, leafy greens, other whole grains, mangoes. So another source of spermidine for the body is through the endogenous cellular bioproduction. So spermidine can be downstream produced from its precursor polyamines, or it can also be catabolized back from spermine. A small part of spermidine is also produced by the gut microbiota. With aging, spermidine production, the endogenous bioproduction is declining, has been shown to decline, and food sources, so nutrition no longer represents enough, no longer gives enough to make up for this loss. So we need to perhaps think about additional supplementation. Now, blood analysis from ImmuneAge has indeed confirmed an age-related decline of spermidine with a strong statistical trend. I forgot to maybe mention earlier that we took 40 participants, right? From each age group, there were 40 participants, which functionally means 20 of participants from each age group in the intervention group after randomization. So practically, a strong statistical trend was observed for spermidine decline in PBMCs. For those of you who don't know what PBMC stands for, peripheral blood mononuclear cells. And we also saw a trend for spermine declining, but unfortunately this trial, the dose and the time of supplementation was not enough to leave marked changes on polyamine dynamics. However, I still want to show you a preliminary result where we saw a trend for precursors declining and perhaps a small indicatory trend for spermine and spermidine increase, which would need to be, of course, verified in bigger numbers and longer trials. So next, since spermidine by itself has been extensively researched due to its pleiotropic effects on age-regulating mechanisms. Among those, autophagy is causally, so it's related basically to regulating aging, and spermidine as autophagy regulator. This was the obvious place to start to measure autophagy, but before I go into the results about autophagy, let me review the process a little bit for you. Most of us already are familiar with autophagy. However, the simple scheme illustrates how this may work in the cell. Autophagy is, I think, safe to say, one of the most crucial mechanisms, cellular pathways for maintaining the health of the cell and longevity of the cell. So you can imagine it as the vacuum cleaner with the shredding function. This simple scheme illustrates in the cell, as the cell metabolizes and ages, there are certain harmful debris that is accumulating, such as misfolded proteins, so aggregated protein complexes, and even whole organelles. And such material is basically sequestered within, triggers the initiation of the autophagosome formation, and triggers the transformation of a protein that we know as LC321 to form a lipidated, so it promotes a lipidation of this protein to form a lipidated version of LC32, which is then integrated into the autophagosomal membrane and serves basically as a docking site for another protein, P62, which then brings into the autophagosome this harmful cargo, which I mentioned. And throughout the fusion with lysosomes in the autolysosomes is where basically the degradation of this cargo happens. Now, the acidic environment in autolysosomes is really important for this degradation, and this environment is basically maintained with a proton pump, VATPase proton pump on autolysosomes, which is the target of this drug that we use in experiments. It's called concanaminase A, which prevents the function of this proton pump and so stops autophagy at the degradation step. With that, we can sort of like pause the autophagy in real time and assess the autophagic flux. Now, in ImmuneAge, we observed the following. So the LC32 lipidated versus the steady state LC32 this ratio has increased from baseline to the final visit, which indicates an increase in autophagic flux in young PBMCs. A similar trend we noticed for old PBMCs. However, this was not yet statistically significant. With a higher dose or maybe longer trial, bigger numbers, we would have also arrived at significance perhaps in old PBMCs. Nonetheless, when we measured the P62, we saw that from the baseline, we saw that basically from baseline to the final, after stopping autophagy at the degradation step, P62 was more accumulated in the spermidine group. Now, I just want to clarify, LC32 is basically proportional sort of with the amounts of autophagosomes formed, while P62 is inversely related to the autophagic flux, so because the protein itself is degraded alongside the cargo. So after blocking the autophagy, and we see an increase of P62, this kind of denotes that some boost in autophagic flux is also happening in the old PBMCs, as we also kind of noticed the trend before. So more autophagosomes and more cargo is being degraded, more material is being degraded. On the other hand, other protein degradational systems, such as proteasomal, were also noted to be generally higher in young cells at all times. And we saw that spermidine from baseline to the final kind of increased the proteasomal activity, but compared to the control group, the proteasomal activity was still a bit lower. This was, of course, insignificant yet, but it does show a trend that perhaps there is there's a crosstalk between proteasomal and autophagosomal activity, and so could be a compensatory effect for boosting the autophagy in young cells. Next, I want to give you an introduction into another process that we studied. It's a bit less known, also perhaps in the scientific community, and has been in the latest years, decades, I would say, identified also as one of the crucial processes and pathways that is regulating aging in the cell. It's perhaps just as important as autophagy. So this is called hyposination, and once again, it shows that spermidine is a direct regulator of hyposination. So spermidine is a direct donor of this aminobutyl group to the eukaryotic initiation factor of translation 5A, which is the only protein that has been so far identified to be hyposinated. Research has been trying to find other proteins that undergo hyposination, but it's only this one. And this process, just like autophagy, is highly conserved between species. So this aminobutyl moiety gets attached to the lysine of the EF5A, and throughout subsequent hydroxylation forms an active hyposine. This all happens post-translationally, by the way, and this hyposine is deserved for regulating translation of several genes, several age-regulating genes. Among those are also mitochondrial and autophagic genes. So in vitro, studies already show that hyposination is very important for the cells to respond to infections, for example, and that because hyposination is also a process which is declining with age, just like autophagy, and spermidine has been shown to restore its flux. So hyposination and autophagy are highly important in the immune system, and spermidine can restore their flux. Now, what we saw in MUNH was again strong statistical trends, and we saw that unhyposinated relative to hyposinated EF5A was declining from baseline to the final in the spermidine group. And we saw that spermidine kind of mitigated this age-related increase of unhyposinated EF5A. But now we are going on to the primary outcome of the study. The time tells me to hurry up a little bit. So we wanted to assess the functional changes in the immune system, and so we quantified the populations of innate and adaptive immune cells with flow cytometry. And so first of all, we stimulated the cells with the spike-derived, so with the COVID-derived spike peptide, and the PP6-5 peptide, which is deriving from the human cytomegalovirus. Those stimulations were for antigen-specific stimulation. And for the positive control, for non-specific stimulation, we used staphylococcal enterotoxin B, which is like a supra-antigen that causes immune-wide, non-specific, robust stimulations, reactions of the immune system. Now, we've got all different kinds of data and a lot of results, but I want to just highlight one key finding today for you, which is on the example of natural killer cells. So natural killer cells are very important for the innate immune system because they kind of surveil the blood for the abnormal cells that lack major histocompatibility complex on the surface and kill them. Now, we found, for example, in the case of the positive control general stimulant, that the old natural killer cells are somehow losing this function, this regulatory function, and so they may respond more significantly with perforin secretion than the young cells. But when it comes to the antigen-specific stimulation, such as in the case of PP6-5, we see that young natural killer cells have significantly higher reactivity and so a better, stronger immune system than the old. During this limited 20 days of spermidine supplementation, we did see, interestingly, that in old natural killer cells, spermidine managed to upregulate, so to preserve the memory responses to spike by the final visit. So this was one of the interesting findings that I wanted to highlight today. Next, to complete the story a little bit, we measured the proteomics. So we did the proteomics from plasma. We wanted to see the proteins that respond to short-term spermidine supplementation in plasma. And what we see is, first of all, by comparing plasma proteome of young versus old people. We identified roughly around 400 proteins, but principal component analysis did not show significantly differential clustering between young and old people, meaning that plasma is not that much different. However, when I did some t-test analysis, I still saw some semantic groups which were significantly higher or more upregulated in the young plasma versus the ones that were significantly higher in the old plasma. The main distinction here perhaps is that in young people's plasma there were more factors circulating that have to do with the adaptive immune system regulation, and in the old people we have many more factors that have to do with the innate immune system. Then when I did the longitudinal analysis of which proteins are significantly affected by spermidine supplementation during this short time, again, from the principal component analysis, I didn't see that there would be much of a difference from spermidine supplementation, but nonetheless, when looking at old people's changes, we interestingly saw that there's a lot of factors related to thrombosis and blood clotting that were downregulated in the course of 20 days by spermidine, which could potentially indicate thrombosis protective effect of spermidine in the elderly. Now, for the young people, there wasn't that much of a change, but we did see a trend. So we did see a direction towards improved lipoprotein metabolism and neuroprotection. So a slight indication for that. Now, this was basically it. I don't have much more time left, but I would like to point out some findings that we saw on the mechanistic research in Drosophila. So in Drosophila, we combined the spermidine supplementation with protein restriction because so far a lot is known about the singular effects of those interventions, but we have big knowledge gap in the effects from combining geroprotective treatments. So just shortly, protein restriction is a reduction of dietary proteins without malnutrition, and it's oftentimes used interchangeably with caloric restriction, which is a reduction of the net caloric intake without malnutrition. So recent research, especially at least from the Drosophila front, is showing that this lifespan extension is not mediated by the total calories reduced, but rather by the type of macronutrient restricted, with amino acids playing the biggest role in lifespan determination. We wondered, since both protein restriction and spermidine are autophagy regulators and they work on similar aging pathways, would combining them be synergistic and positive, beneficial, or would it be unnecessary or even occlusive? So from many readouts, in our latest paper, from many readouts, we saw indeed a synergistic and additive effects from combining those treatments. For example, on the lifespan curve in Drosophila, if we look at the green and blue, so blue being the protein-enriched diet versus protein-reduced diet, we see that protein restriction by itself is lifespan prolonging, but so is spermidine, and spermidine works on top of both diets. When we apply it on top of both diets, it did significantly prolong the lifespan of Drosophila. We see similar improvement in locomotion, so in preserving this age-related decline in locomotion on top of both diets. So, and interestingly, as another readout, spermidine also boosted the egg production in Drosophila more significantly on top of the blue protein-restricted diet, but it also worked up until the old age on the protein-reduced diet as well. So synergism in this case may work because a lot of research is showing that those interventions operate via parallel pathways in the cell to regulate similar outcomes, so similar pathways, similar age protective mechanisms, which I don't have the time to go into details now. But to finish this presentation, to wrap it up, if you don't remember anything from what I said today, I guess the points I was trying to make were I wanted to show you what a difference mechanistic research can make in recognizing the facts, or in really having this foundational understanding of the aging mechanisms and where to start. And it's also really important for the future for guiding this precision medicine or future medicine. So I wanted to illustrate spermidine as the adjuvant for immune system related challenges, and I show you ImmuneAge as the first in vivo clinical trial that touched this aspect and that researched on spermidine in this context for functional changes, for functional improvement for the elderly, especially in times that we were facing in 2020. Then I highlight the need for exploring higher doses in spermidine-related trials and bigger numbers of participants. And I wanted to maybe highlight this introduce this importance of studying the combinations of geroprotectors in the future to so target as many aging hallmarks at once. I think this is where the future lies now. So at this point, I would like to thank you, and I think for the major part today, the health still kind of lies in your hands until we have more evidence and better approaches. But I think with this right now, I would be happy to close this dialogue and close this monologue and open a dialogue for questions. Yes. Thank you.
Is peptide 65 a separate agent, or is that part of spermidine? Because it also increases the level of IGF.
Excuse me, could you repeat?
Is this peptide 65 a part of spermidine, or is it a separate agent?
Oh, this is a peptide that is deriving from the virus. It's a viral peptide that's used for stimulating the immune cells.
It's not part of spermidine.
No.
But one of those ingredients, because it's natural food, right?
Yeah.
What are those ingredients for the liquid?
Sorry, I don't hear you acoustically so well. So you're asking if...
What are the ingredients in the spermidine?
Spermidine is a singular molecule. This is a singular agent for itself. It's a molecule that belongs to the class of polyamines, and it is naturally present in all the foods we eat. It's basically, you have it in all of the cells of your body. Without spermidine, without the polyamines, the cell is not viable, so we wouldn't exist. So this polyamine is highly represented in some of the food types, like I mentioned. So wheat germs are the huge source from which spermidine is also getting extracted for supplements, for example. Then we have other foods like mangoes, oranges, fermented soybeans. For example, if you look at some blue zones in Japan, so there are this, I think in Okinawa, for example, the traditional dish of these people is natto, which is made from fermented soybeans. And so these people consume a lot of spermidine. There has been also clinical studies with these people. On the other hand, we have Mediterranean people who eat this Mediterranean diet that is really enriched in spermidine, and these people consume on average up to 30 milligrams of spermidine per day, which is kind of, in comparison to Europeans, would say but between 10 to 15 milligrams, and in comparison to Americans, about 8 to 6 milligrams per day.
And you measure that how, spermidine?
So with this, I think they measure it with liquid chromatography. With there's a special technique for that. I am not so familiar with this because my collaborator institute did that, but it's not like a straightforward way to measure spermidine. You have to really have the lab, and it's nothing that you can just do as a diagnostic on the go.
Oh, I get it. And the last question is, what actually increases the IGF level?
So practically, if we are looking at, if we're talking about the insulin IGF pathway, so this is all factors that contribute to anabolic pathways in the cells, such as overeating or high caloric food that would signal the high energy state in the cell. So, for example, protein restriction would reduce this pathway, and this is
increasing the IGF levels is good.
No, it leads to insulin sensitivity, to decrease in insulin sensitivity with age.
Okay.
So that's why one of the reasons.
There are more IGF levels. Okay, okay.
No, it's fine.
No, it's okay.
Sorry, one question, can I?
Yeah.
Do we have evidence that this spermidine affects health span or lifespan in the human population?
So when it comes to clinical trials with humans, it's difficult because you would need to monitor throughout different generations. So you need to monitor one generation completely. Clinically, this is sort of close to impossible to do, but there has been...
Exactly, this is what I thought.
Yeah, but there has been one observational trial. It's called the Brunex study. It's Italian population of people. It's a population of Italians who on average just naturally eat a lot of spermidine enriched foods versus the ones who don't. And there has been, I think, about, was it 5.7 or 7.5 years of a all-cause mortality difference between these groups. I can share the paper with you later, but there has been this is the only observational trial that we have. And it makes a big difference. So this all-cause mortality was significantly reduced in spermidine eaters. Yes?
You said that the maximum spermidine supplementation in the study till now is like 6 milligrams per day. But what happens if you increase the dose? And will the higher dose be even more beneficial?
All right. Quite common question. So, ImmuneAge was in 2020-21, and this was by this time the highest used dose. Now, just today in the morning, I opened a study that studied 40 milligrams of spermidine. But now, and then in between, there was another study with 15 milligrams per day. But at the time, we had limitations because we had the spermidine from this company that was packing spermidine in these sachets of 2 milligrams per sachet. And for the compliance reasons, we couldn't increase the spermidine to so much because, people had to drink these sachets like 3 times a day to get these 6 milligrams with every meal. And so now with the upcoming, or I think they are already in use for clinical purposes, like synthetic spermidine, we will have the capacity to pack much more spermidine in capsules, and so we are able to really increase the dose in clinical trials. Now, of course, if we had increased it in ImmuneAge, we would have seen many more robust outcomes in this trial in these 20 days if we had used higher dose, but also if we would have done it for longer. Our point was to basically prove if, like before some immune system related treatments or interventions, you could just do the spermidine for like short time before that, and this would really help you with like immune system reactions to either vaccinations or to infections. And so, the benefits are there. The safety and tolerability before this trial was also assessed in 3 months study. There is generally very little doubt about spermidine being safe. There has been no adverse events reported. There are some question marks, which I can address as well. But in general, spermidine seems to be a very safe substance. Yes?
Sorry. No,
It's
just quick. Yeah.
very quick. Thanks,
Sure.
Ron. First of all, I just wanted to say thank you so much for your research. First of all, I really appreciate that you're doing hard scientific research on spermidine, because I've been hearing about it for the last several years, I would say, on what I'll call the longevity circuit, along with things like rapamycin and metformin. And of course, we don't have 90-year clinical trials, which is what we would need to answer the gentleman's question, really, right? So first of all, I wanted to thank you. And then second, I wanted to ask you, what do you need to continue your research? Is it money, encouragement? What do you need?
Oh,
So much everything.
Thank you. Wow, I would need a team, a lab. So since I'm just finished my time in my former lab, so I would need to be incorporated again in a team. Then, so I would need all of those that you listed. I would need time. I would need like efficient cooperation, because throughout my clinical trial, I saw we had, unfortunately, my clinical trial just fell in the time of COVID, so we had a lot of restrictions, and still I managed to pull it off with kind of like nagging, and you just have to, as a doctoral student, sometimes, you're not the priority on everyone's list. And I would need the efficiency. So the teams that really work efficiently and on point, and that we can avoid these unnecessary scientific logs, which are just, there's a lot of them, and science gets really slow as it is already. Definitely money can help with that. But so can facilities, resources, a lot of expertise, for example, people with different kinds of expertise. So all of that, practically. And I see that somebody from the Zoom.
We have one question. We will say one question,
of course.
And then we are over the time.
Yeah, and then maybe I just.
Do we have a break right after this?
Yeah.
Great.
So it's okay.
I'm willing to stay to hear the question during.
Oh, cool. So then.
Okay, one question for the Zoom.
Let's process the.
Yeah. So the question goes: which food is the richest in spermidine?
Okay, well, go to the bio store and buy a pack of wheat germs and put them in yogurt, in soups, in whatever you feel like, and just eat a lot of these wheat germs. But also, don't eat only wheat germs. So the truth is double-edged. So first of all, if you are a disciplined person, and if you are a consistent person, you can just change your dietary habits, and you're going to ramp up on your spermidine naturally. And if you're not a disciplined person, by all means, for consistency, and if you have enough money, go and buy the supplements. So there are two ways to do that, right? There are, interestingly, some studies that are showing benefits of spermidine from diet directly, but they didn't see some other independent studies with similar doses did not really see effect from supplements. So there is this lingering remaining question of whether spermidine is easily and better metabolized from food sources rather than from the supplements, because now in supplements we still mostly have plant extracts from wheat germs, but soon we're also going to have like a synthetically produced spermidine, and the bioavailability of polyamines from these supplements is kind of still questionable, because we don't have a good clinical trial demonstrating the effects from the supplements versus food. And generally, we have this knowledge gap in polyamine distribution and dynamics in bloodstream upon supplementation. That's what we don't have. So in ImmuneAge, we addressed that. I didn't show you, we quantified polyamines throughout all blood fractions. But in this 20 days, we didn't see any significant difference like that spermidine would change the polyamine dynamic in the bloodstream so much. So this question still remains. But yeah, I hope I answered the question from the Zoom audience. And I would need to stop here because they're urging me to get off stage. And I'm happy to talk in the break. And I'm also happy to take all of the questions from the Zoom. If I can answer them anyhow, if people... But in any case, thank you very much for having me.